topic of biology through the eyes of an engineer. I've known Parvez for the last 4 years or so. And Parvez is currently a senior lecturer in materials modelling and design at the University of Edinburgh. He's a fellow of the Royal Society of Biology, a fellow of the Institute of Materials, Minerals and Mining. He's a chartered mechanical engineer and is a member of the Institution for Mechanical Engineering. To say that Parvez has a varied background is an understatement. Parvez did his bachelor's degree at the University of Bath in materials science and followed that up with a PhD in 2004 in mechanical engineering, but in a topic that I would suggest is perhaps more civil engineering, which, if I remember right, Parvez, was the design of a timber truss based on the structure of a dragonfly wing. Is that correct?
Oh, no, that was later. That was later. It was,
okay.
Timber beam reinforcement with FRP.
Okay. So after that, Parvez traveled to Finland, where he was a researcher at Abo Akademi University in Finland. And he rose from being a researcher to being a principal investigator and group leader. And then he came to Edinburgh in 2016, where he was a senior Marie Curie fellow working on composite materials for marine renewable energy. And in 2018, he became a senior lecturer. Parvez, no doubt, will tell you about a lot of these things, but he's also a visiting scholar at Gajah Mada University in Indonesia. So with that, Parvez, if you'd like to share your screen with us.
Sure.
And take it away.
Okay, let's find this quickly. Okay, I'm sharing my screen now. Can you see the screen?
Yes.
Excellent. And I guess you can hear me, so let's just kick off then. All right, well, thank you very much for the introduction, Kahir. That was a really nice one. And thanks also for the invitation to give a talk here today. Okay, so I'm just wondering, how do I move these forwards? Okay, found it. Basically today's talk is entitled Biology Through the Eyes of an Engineer. I'm really happy to see that there are, a lot of people who've signed up for this, and I hope you guys enjoy the presentation. So before I started preparing this presentation, I started doing some Google searches. I started wondering to myself, how many people are interested in the similarities and differences between biology and engineering and science and engineering and so forth. And it came to my attention, there's actually quite a lot of people who, who are actually asking this question on the internet. And one of the better sort of answers, I think, comes through Theodore von Karman's statement, which was, scientists study the world as it is, engineers create the world that has never been. And that kind of goes hand in hand with the etymological root for the word engineer, which basically comes from two Latin words, one meaning create and the other one cleverness. But let's just go back to Theodore von Karman's statement. He was a physicist and an aerospace engineer. He says, scientists study the world as it is, engineers create the world that has never been. But what if you're one of these awkward people like myself who wants to study the world as it is in order to recreate the world as it is? Well, I guess those guys need to fit in somewhere, and biomimetic design, bio-inspired science, bioutility, these areas in general are quite descriptive, I think, of where those guys could fit. And when it comes to biomimetic design, I've pretty much split this up into four separate sort of sections, four areas that are interconnected, but all equally important, and most importantly, are all equally fun. The first is obviously exploration and discovery. You need to go out, you need to search, you need to find, you need to, basically discover all these special organisms that will give you some inspiration for engineering practice. Then, of course, you need to do the research to find out how these things are working. And I'm just terming it bioresearch for now. Following that, I think mimicry and manufacture comes next. And that's pretty difficult, I think, in many cases. And I'll get on to that and reasons behind why that's difficult later towards the end. And then, of course, one needs to start applying these things to technology in order to do the full, to complete the full circle, so to speak. Now, there's always a story, and what I'm going to do today is I'm not going to go chronologically from part to part. What I'm going to do is I'm going to basically merge all of these together and just tell you a few stories in between. And I hope you enjoy the stories, and I hope you enjoy the kind of research that has been done over the years. So the first story I'm going to start with is it originates in East Africa. I heard a rumor that a group of mothers in Kenya had been farming spiders and then using the spider silk to make like different types of products, like different textiles and so forth. And I thought to myself, well, this rumor sounds kind of crazy, because all the scientists will tell you that you cannot farm spiders, because spiders are inherently cannibalistic. If you put them in close proximity to one another, they'll just basically eat each other. So I was thinking to myself, well, I travel a lot and I see a lot of crazy things. So, why can't this rumor be true? So I decided to go to Kenya. I got in a car with a couple of my Kenyan friends over there and just started traveling around following this rumor. This is not the specific route that I took. I can't remember the specific route that I took, but we did travel for quite a long time, and eventually we ended up there in Kisumu. And when I got to Kisumu, that's where this rumor came to an end, because I got to this, the place where the rumor was meant to have originated, and it was a silk factory, but it was a Bombyx Murray silk factory. So that's your typical silk that people have been using for thousands of years. And obviously I was not feeling too great at this stage, but I do stand out like a light bulb in Kenya. And that's when I found these guys. I was there outside the silk factory, or I should say these guys found me, because they came over to me and they were saying, what are you doing here, Muzungu? Yeah. And for those of you who don't know, Muzungu means white man. And it was probably the first time in my life I was called a white man. But anyhow, I was okay with that. I took it and I was just like, well, basically, this is what happened, and, I'm feeling very sad, a bit depressed, blah, 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 because we traveled so far and we didn't find, the rumor to be true. And they were just there, yeah, we can do this because we're insect farmers. And, you don't find that many insect farmers in Kenya. So I went to see their insect farms, and they had all sorts of insects being farmed in different mud huts and stuff. And basically, I thought to myself, well, these are the guys then. And I thought to myself, maybe this is how the rumor originated. You had insect farmers living in close proximity to a silk factory, and voila, you've got your rumor. However, we basically decided to work together. I helped them establish a company, Kisumu Insect Farms. And that's basically when we spent 2 to 2.5 years bioprospecting for different spiders and trying to understand the properties of their silks. And then we found this one, Nephilingus cruentata is the name of this one. And it basically spins an egg case that has a toughness, not the egg case itself, but the threads in the egg case, that has a toughness of 193 megajoules per cubic meter. Now, for those of you who are not familiar with toughness terms and values, 193 megajoules per cubic meter was number one, 30 megajoules per cubic meter higher than the previous record holder, which was, I think that was in Italy that they found that spider. And number two, if you think about it like this, steel, depending on the type of steel you're using, will have a toughness of between 1 and 5, typically, megajoules per cubic meter. So these silk threads, which are polymers, whereas steel is metallically bonded, this is a polymer, is around about 100 to 200-ish times higher in toughness. Another thing that this egg case had was a ripstop architecture. I could not put my fingers through it. I could not put stuff through it. I eventually cut it with a knife and then I just put my fingers through, and I'm a fairly strong guy, right? I put my fingers through, and I tried to rip this spider egg case, and I could not rip it open. So it seems to have this ripstop architecture. When you pull it open, the threads around it seem to tuck in, and they close in. And this is something that I'm planning to work on a bit more with one of my colleagues, Vasilios Koutsos, at the University of Edinburgh. We're hopefully getting a PhD student in within the next year, let's say, depending on the COVID-19 situation. Anyhow, the egg cases are specifically tubular form silks, right? Or sometimes they're called cylindriform silks. And they're similar in structure and properties to dragline silks. There are 7 different types of silk. I'll speak about that in a sec. But basically, how is it that these polymer materials are so tough? I mean, that is, I guess, the question that we have here. Well, one of the reasons that they're so tough is basically that they are nanocomposites, and they have these polyalanine segments that during the spinning process, the spider will spin, and then it'll sort of basically extrude this silk from its spinnerets. And the rate of extrusion will align the molecules, and through molecular alignment, you'll have polyalanine segments lining up and creating huge numbers of hydrogen bonds, which then end up resisting shear and keeping, storing energy within the nanocrystal. At the same time, you have this semi-amorphous matrix, which is made up of random coils and, alpha helices and that kind of stuff. And so you've got this soft matter around these nanocrystals. So you have both stored energy and you have stretchability of the silk. Now, obviously, in nature, optimization is function specific. It's loading specific as well. So the silks, this guy here, he's saying biomimeticist dreamers have no chance while standing next to the Eiffel Tower, and he's absolutely right. If you were to pull this in tension, no problem. You're basically utilizing that silk in the manner that it has evolved for. However, if you were to squash it in compression or try to bend it, it wouldn't be so useful, right? So while Tarzan would have loved a material like this, somebody like Vlad the Impaler, may have questioned its immediate benefit. Anyhow, I did mention that I was going to speak to you about the different types of silk. There are 7 different types of silk that spiders can spin. The egg cases are cylindrical or cylindriform, tubular form silks. You also have sticky glue-like silks called the aggregate and piriform silks. And of course, you have the drag line, the really strong, and the tough silks like the major ampellet silks. But there's also a silk called the flagelliform silk, which forms the capture spiral thread. And these are very spring-like. And this is an interesting silk. All of the silks are actually very interesting. This one, when we modeled this silk, we noticed that actually, even though it is not traditionally seen as a bioglue, whereas the piriform and the aggregate gland silks are, this particular silk has, a spiral structure that forms a huge number of intramolecular hydrogen bonds. Whereas in the dragline and the tubular form, you've got intermolecular hydrogen bonds. These ones are intramolecular hydrogen bonds. And so what we thought is that if we were able to identify and isolate the amino acids that are actually responsible for mass hydrogen bonding in the flagelliform silk, we could use monomeric forms of these amino acids as bioglues and apply them to stiff natural biopolymers like nanocellulose. And the best one, in my opinion, to use is bacterial nanocellulose. So it's a very difficult word to say for some reason. Bacterial nanocellulose, because it is 98% pure, and it doesn't have, the lignin and the other sort of impurities in the same way that nanocellulose from wood or from tree, woody sources has. So we did that, and we actually managed to double the strength with respect to density and increase by 5-fold the stiffness with respect to density. So the specific strength and specific stiffness respectively, as compared to normal bacterial cellulose sheets. And they are, of course, then composites, because the monomeric amino acids form essentially the matrix, or the bioglue that holds together the fibers. Now, whilst modeling the internal structures of silk, one of the discoveries that we made through the modeling that has led on to this hypothesis that we need to, test experimentally, is that semi-crystalline regions form at the interface between nanocrystals and amorphous matter. And this seems logical, actually, from an energetic perspective, because you've got the amorphous matter that's basically crumbling and the stiff matter that's trying to hold itself up. So logically, at the interface, it makes sense that it would form semi-crystalline regions. And semi-crystalline regions in silk have been identified, but we just don't know where exactly they are. So it would be nice at some point to do some NMR and some Saxwax work on this, just to try to better figure out, if this hypothesis that has been generated from the modeling is in fact true. And this hypothesis stands also for over-sheared silk strands as well. Now, spiders are not the only, as I mentioned before, they're not the only organisms that produce silks. Some snails do as well, but this is probably the most famous silk producer. This is the Bombyx mori moth. And on the right-hand side of the slide, you will see the cocoons that basically are degummed of sericin, which is a sticky glue, to leave the structural fibers known as fibroins that are used then to make your silk clothes and so forth. But, I got my hands on these cocoons, right? And I thought to myself, these are really stiff and really strong, but they're really useless and impractical for me in this format, because look at the shape, right? So I thought to myself, there's so much sericin wastage on the earth. Why don't we degum these cocoons, right? Degum them, then take the structural fibers, the fibroins, and then straighten them up. So we've got unidirectional fibers, and then regum the fibers using the sericin that we degummed earlier. Now, this is very interesting, because we did this and then we tested them, and we got this really, really interesting result here. This is not one result. There's obviously repeatability. I'm just showing you representative curves, but we do lose about a third of the strength if we do that. Quite a lot of stiffness, but we do massively increase the strain to failure to 200%, and we do massively therefore increase the toughness, or the mechanical energy absorption that is possible with this kind of material. Now, we had that. Could you turn your microphones off? Thanks. Anyhow, I'll move on from there. So that's the silk story. And we all know that these spiders, they also attach, they also climb. And I'm not going to speak about spiders climbing and spiders attaching. I'm going to speak about other organisms climbing and attaching instead. And I'm going to start this section with a quote from Albert Einstein, who said, "Everybody is a genius, but if you judge a fish by its ability to climb a tree, it will live its whole life believing that it's stupid." All but this fish. This fish is Periophthalmus variabilis, and we came across this fish in Semarang in Indonesia several years ago. And it's, it is quite an interesting fish, just because it does climb trees. There are quite a few terrestrial fish out there. But this is the only one that we know of that climbs trees. There is another one in Bangladesh, actually, that can kind of climb, but it uses spikes under its legs. But these guys, they can climb really fast, and they're very adept at climbing. And they spend a lot of their time just sunbathing in the trees and so forth. And it's a weird sight, when you're in the mangroves and you see these fish on the branches in trees sunbathing. Anyhow, what I needed to know was, how come they are able to climb trees. And I thought the best way to try and figure that out was to compare them against fish such as BIOMIMETIC species, because these are also terrestrial fish, but they are merely able to, merely able to crouch and crawl on land. So what we decided to do was compare certain physiological features that were clearly different between the two. And one that stood out was the pelvic fin. The pelvic fin of the land-walking species, I'll call them for now, is fused, and it also has a specific structure with lots of fin rays that base on top of the fin web. So the fin web is flexible, the fin rays are quite rigid. And it seems that that lack of flexibility as a result of the fin rays is possibly why it's unable to stick well to surfaces. All of these fish secrete hyaluronic acid, which is a very sticky biopolymer. But the land-walking fish don't seem to be able to use those when they start going onto vertical or sloped surfaces. Whereas the tree climbers, the periophthalmus species, they do. They actually have this structure you see on the right-hand side there, where their rays are organized in such a way, and there's less of them. So organized in such a way that you have less of them, well, less in terms of fraction relative to the fraction of web, such that there's a lot of flexibility that's possible in the pelvic fin. And we've seen for ourselves these pelvic fins, they actually, they attach to the surfaces. You have capillarity, you have Stefan adhesion, and so on and so forth. And basically, these fish are able to climb as a result of at least the functional morphology of their pelvic fins. Now, as if it wasn't weird enough to see fish sunbathing in branches on trees, we then noticed that some of these same fish, this specific species, P. variabilis, that they can also fly while hopping on the water surface, right? And I'm going to just play you this video here. Watch very carefully. You see the stick pointing in from the right is going to disturb this, this fish that's hanging out on a mangrove root, and it's going to hop its way across. So check it out now. There it goes. Off it goes. There you go. So it taxis on the water surface, and then it flies. It basically hops on the water, and it prefers to do this than swim underwater and swim away, which seems crazy to me, but it does this pretty fast. Anyhow, 3 years later, several visits to Indonesia later, and lots of video footage later, and kinematic and kinetic analyses later, we have managed to characterize the way in which this this fish actually hops and so forth. And it is a completely new form of locomotion, fish locomotion that has never been seen before. The flying fish is different because it taxis for long periods and then it glides basically. Whereas this guy, he taxis short bursts, taxiing on the surface of the water, and then it hops, and you have this sinuous motion in the air as well. But the best thing when it comes to these kinds of projects, besides obviously going out into doing the field work and spending your entire day chasing fish, the best thing besides that is interacting with our students at Edinburgh and bringing these projects to them. And this year we've got a group project where our students at Edinburgh are basically using these fish now to inspire the development of multi-terrain robots. And they're obviously, because of the pandemic, very sadly, they can't make these robots physically this time round. It says Edinburgh UGS there, by the way. That stands for undergraduates, so it's not meant to be OGs. But anyhow, they're basically designing robots this year round. And it's really, really fun because, a lot of these projects that I bring over to our students are, kind of off the wall in some ways, but they still need to use their engineering skills in order to be able to develop robots and so on and so forth. And the benefit of having a multi-terrain robot, by the way, is of course for things like rescue operations and so forth. So there is an application purpose ultimately behind it. But, you know, the number one most important thing, of course, in my opinion, is that the students are enjoying the projects. And this is another project that, you know, that the students undertook after I went over to Australia for a while to work with Christopher Clement on tree climbing lizards and burrowing lizards. So basically, what we wanted to do is understand whether there are morphological differences in the claws and the, you know, of arboreal or tree climbing lizards as compared to burrowing lizards that gives them an advantage when it comes to climbing. Because if we understand that, perhaps we can apply that technology to biomimetic robots. Now you might say, but don't we have climbing robots? And I'll get to that in a second, because there are some interesting findings that these students came to during the course of their project. And this is one of the group projects from last year. So basically they did some morphometric analyses of the different types of claws, the claw shapes and so forth. They did 3D scans, 3D printing, and then they you know, developed models, penetration models, and they, you know, they had to basically design and develop their own testing apparatus, because of course there aren't standards out there for testing lizard claws. So they had to make their own apparatus to standardize the tests between the arboreal and the burrowing lizards. And what they did, everything they did led to the same you know, affirmation of our hypothesis, which was that there are certain morphological features in lizard claws that climb trees that benefit climbing over the claws of burrowing lizards. And basically, one of the things that they found through this work was that the arboreal lizard claws actually worked better than the claws for the climbing NASA robot called LEMUR, right? And if you look at the difference between the arboreal claw and the LEMUR claw, which is, now I do understand that NASA have a lot of fantastic engineers. I'm not saying anything against NASA. And of course, these claws on the LEMUR, they're arranged in a circle, so they've got all of their degrees of freedom covered. However, if you were to take individual claws and compare them both, the NASA LEMUR does not work or does not function, does not grip anywhere near as well as a tree climbing lizard claw. And that is ultimately down to the fact that it's got flat sides, whereas the lizard claw has very sharp curvatures. Okay, so I'm going to move into another student project that's ongoing this year. However, I'm going to speak to you about a little bit about the background behind this project. So a few years in a row, I've been trying to get cicadas from Indonesia, because cicadas, they're really, really interesting creatures generally. They're these fat bugs, and, you know, it depends where you are in the world. Some cicadas, you know, they will come about annually. Some cicadas, you know, they will appear like every 2 years, some of them every 7 years, and so forth. They're very similar in that sense to dragonflies. Dragonflies in the equatorial region, for example, they will stay in a larval form underwater for 1 year. Whereas if you go to the north of Finland, where it's super cold, they will stay in a larval form for 12 years. So, you know, cicadas are, in similitude to dragonflies, are quite precious bugs in that sense, because you know, depending on where you are, you may not be able to get them very regularly, and certainly you won't get them throughout the year. So we conducted several expeditions searching for these cicadas, and sadly, you know, every time I was on expedition, we did find them, but we did not manage to catch them because they are super fast. And obviously, you know, on some of the longer expeditions, you get hungry and you end up eating their cousins. In this case, the crickets that were baked here. But basically, our purpose for capturing these cicadas, we had different purposes. The one I'm going to speak to you about today relates to stored energy in the wings. And there is an interesting story as to how we eventually got our cicadas, because the students from Indonesia who were coming to Edinburgh, they had the tickets booked, everything was set. They were going to work on cicadas and crabs. We had the crabs, we didn't have cicadas, right? So, you know, about a month before they came, and I was fairly sure they were not going to work on cicadas because we had none. A month before we came, Puspa, who is one of the students who came, she opened her window and a cicada flew in. So we had one. And then the next day she did the same thing and two flew in. And she kept doing this for about a week, and then eventually we had about 14 cicadas that were captured, and then we just quickly got the permission sorted so that they could be transferred to the UK. But one of the things that we want to study with these cicadas is stored energy in the wing. Yeah. So for the upstroke and the downstroke, I'm going to just play the top left. It's difficult to see because this is a 2000 frame per second camera. It's a black and white one, but hopefully you'll be able to see. Do you see it springs back there? And then if you look at the bottom one, that was the upstroke. This is the downstroke. You see it springs down for the downstroke. So at the maximum pitch angle of this wing, right, there is elastic stored energy in the wing. And one of the things we hypothesize is that perhaps the cicadas save energy. You know, they don't use their muscles so much to move their wings because they have a lot of stored energy in their wings that, you know, basically allows the wing to flap back. And it does so at quite high speeds. We check this on several different specimens, you know, the 14 that we had, and they're showing the same kinds of results. That for the downstroke, we're getting up to 21, 22, 23,000 degrees per second of motion. And for the upstroke, it's a bit less, it's about 11,000 degrees per second for the upstroke. As you can see, these curves, you know, they're kind of, you know, the downstrokes are skewed bell-shaped, but the upstrokes are fairly symmetrically bell-shaped as well, which is kind of what you would expect for a wing that's springing back. So what we thought we could do is try and design robotic wings for the undergraduate students this year that could make pollinating robots more energetically efficient. And these guys are this year's group who are working on this, and they're using different types of kinematic modeling and finite element modeling techniques to redesign, and material selection techniques to redesign wings so that they can spring back as energetically efficiently as possible. And, you know, of course, there's a lot more that can be done. You need to couple this ultimately with, you know, the aerodynamics of the system as well. But, you know, one step at a time. And most important thing is that the students are learning new things and that they're having fun. Anyhow, since we're speaking about cicadas, another flying insect that I've had a lot to do with is the dragonfly. And one of the things that puzzled me for a long time with dragonflies is we got these dragonflies with really thin wings that are made predominantly of chitin, and chitin is a brittle biopolymer. So you got these wings made of chitin, they're super thin, and they're flapping between 6 and 30 times per second, which is not actually the fastest flapper in the world. The fastest flapper in the world is local to here. The fastest flapper in the world is the Scottish midge, which can flap over 1,000 times per second. By the way, that's just a short interlude for, you know, for your interest. But anyway, 6 to 30 times per second with a wing of this size is still a lot of cyclic loading on a wing. And you know, I just think to myself, why isn't this thing tearing? Why isn't it breaking up? Because it makes no sense, right? So what we did is we analyzed the, you know, optically using SEM, using light microscopy, and also confocal microscopy as well. We kind of analyzed the structures and found that in the joints of the wings, there was a hard, soft, hard segmented structure. And the soft part was resilin, essentially. It's a very rubbery protein biopolymer that these dragonflies produce. And it seems that the resilin deforms a lot during flight, and that it basically stores a lot of that energy and then it just releases it. But as a result of it being soft and being able to deform a lot, the wings don't actually endure very or highly localized strains at interfaces, which means that they're less likely to fail. And I thought to myself, and this is something Caher mentioned at the beginning, I thought to myself, why don't we try and apply this to something in engineering? Why don't we try, since I have a PhD background in timber engineering and timber composites, well, why don't we just try to improve the fatigue performance of bolted timber truss joints, right? So we just, we drilled some holes into wooden trusses, bolted them, and then we did the same thing. But we put a layer, 2.5 millimeter thick layer of rubbery glue around the bolts in the second set of tests, and we actually ended up doubling the fatigue lifetime of the trusses. So there is potential. That was preliminary work. There's much more that can be done there. Anyhow, I'm going to move on now to a different arthropod. That's a flying arthropod. This is a marine arthropod. And this one is called a snapping shrimp, a pistol shrimp, right? Basically, this shrimp has a massive gun on one of its arms. And this gun has a plunger socket system that works like this. And it basically squeezes air in this plunger socket system, and it's shot out through this gun. And it basically this cavitation bubble is shot out, and then when the cavitation bubble collapses, it reaches the temperature of the surface of the sun, so about 6,000 Kelvin. Now, obviously, energy created, destroyed, and so forth. Well, there's surely a lot of pressure and temperature in that plunger socket system, and I'll show you the plunger socket system here. You see at the top there, plunger and socket. It's basically doing that. So there's surely going to be a lot of energy and pressure and temperature arising within that plunger socket system. Though we haven't actually measured it ourselves, it makes sense that there would be. So what we decided to do is try to characterize and understand what it is about this plunger that allows it to survive. If when you're trying to capture these things, right, they start walking back like this, and then they start going bang, 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 and they just keep shooting you with their gun over and over and over again. And it's like, for you to apply repetitive cyclic pressures like that into this system, it's got to be a fairly robust system. And if there are high temperatures developing in there, then it needs to be able to circumvent those temperatures so you don't burn up the tissue inside the cella. But basically, we cleaved these plungers, and what we found was that they are composites. They're sandwich composites. So you've got a bottom layer and an outer layer and an inner layer that are made of a composite itself, which is a calcium carbonate, like bonded by proteins and polysaccharide type matrices. And then in between those, you've got this. If you look at images C and D in that slide, you've got this layered Christmas tree porous structure that is actually made out of chitin. So basically, what's happening here is that the heat, and we modeled this, and you can check the paper, is really interesting. The model is that the heat, it can go through the first layer, that's the hard layer, right? That's probably good for pressure. But then the heat does not go through this middle layer, which is this laminated Christmas tree-like porous chitinous structure. It's very interesting stuff, and there's a lot of possibilities with a structure like that when it comes to aerospace. For example, you could be, maybe you want to retain heat, or you want to deflect heat or something like that. If you want to retain it, then perhaps it could be useful in space. As a thin film on suits, it gets to minus 270 in space, I think, something like that. And if you're on the top of a space shuttle, was it 1,650 degrees Celsius as you re-enter the Earth's atmosphere? So there are possibilities if we can manufacture things like that. I'm going to get to that again. I'll mention the manufacturing stuff a bit later. Now, other marine organisms that we have studied, there are many, but other ones that we've looked at are crabs. And we've looked at crabs in several different ways and from several different angles. recently, we looked at these 5 crabs that were collected by the Marine Study Group from Gajah Mada University on Wangi Wangi Island in Indonesia. And there are 5 different species of brachyuran crabs. What we did is we basically separated the shells from the crabs, removed the insides, cleaned them up, and then filled the insides with silicone in order to mimic the body structure. You don't really want to be doing cyclic impact tests on crab shells when the original body is there. Or maybe you do, I don't know, but I certainly didn't want to be involved in something like that. Anyhow, importantly, the crab shells, they do fail in very different ways. Some of them are more brittle and some of them are, tend to show more ductile denting characteristics. And you can see that iterated again here through this graph. You've got L. sanguineus and P. vespertilio that show ductile characteristics. We've got the residual stiffness here plotted against the number of impact cycles, whereas the other 3 crab shells seem to be quite brittle in their, in their cyclic failure character. So in order to better understand how shape affects, or shape potentially could affect the failure mechanisms, I asked my PhD student Gabrielis to basically run some simulations on the 3D scans we had of these crabs, impact simulations, and we got quite a lot of interesting data out of that. One of the things that we did notice is that while there were mild correlations between different morphological features of the crab shells, such as the arc length, the thickness, the carapace angle, the topographical irregularity, and the Wenzel roughness, while there were only mild correlations there were very clear correlations when it came to stress peaks, where there were valleys in the deep valleys in the crab shell. So basically, the deeper the valleys were, the higher the stress peak. And since crab carapaces are brittle materials, it follows that if you have a stress peak in a valley, or where there is a geometrical change on the carapace surface, that you'll probably end up, having some issues. Sorry, there was a message that popped up. I don't know what that said, but let's just continue. Anyhow, so in summary, when it comes to that, crab species with brittle failure characteristics exhibit both the greatest arc lengths and the deepest V grooves. Whereas if you want ductile denting failure modes of a brittle material, then you need to have shorter arc lengths and smaller, more broadly distributed carapace grooves. And these are very different to corrugated plastics, which are ductile materials. Corrugated plastics will work well in impact because they can deform a lot, but these are brittle materials. So the application areas are different. These, you could get inspiration for application in dental molds, contoured military armors, and that kind of stuff when it comes to these types of structures. Now, that's the outside of the crab, the geometry that relates to the outside of the crab. When it comes to the inside of the crab, there are also impact-resistant geometries. And basically, inside the crab shell, you have, and this is true also for the mantis shrimp dactyl club, and also for the beetle shells as well, you have Bouligan-structured composites. These are fibrous, laminated structures that basically twist like a twisted plywood structure over and over again. For each subsequent laminate, it twists at a slightly different angle from the previous one, and it twists like that. And basically, if you were to smash a structure like that, then the fracture path would have a much more tortuous journey to make than if it was a unidirectional composite where that fracture line could just work its way straight down. So due to the increased tortuosity as a function of the geometrical architecture that these fibers are making, you have impact resistant shells. And one of my PhD students, as you can see, this guy here at the top, Chidume, he's working on developing improved impact resistant properties of carbon fiber reinforced plastics. And he's basically, at the moment, he's obviously access to the lab is limited at the moment, but he's done some work on DMTA, and he's done some work on tensile testing of these kinds of structures, and now we're looking into hydrothermal aging of them as well, and so forth. So there's a lot of potential with these kinds of structures, and there are others who have studied them as well. Anyhow, since I'm speaking about composites, I'm going to just switch to another organism altogether, the sperm whale. I did a lot of work on sperm whales and baleen whales in South Africa several years ago with Professor Anusia Chinsami Turan. And basically what I wanted to just jump in with here is, since I'm speaking about composites, is that the sperm whale skull amphitheater, that's the amphitheater, is that crest at the back of the head that you can see. It's basically an extended occipital bone, and that extension it forms this crest. It looks like some of the old dinosaurs, right? Anyhow, it is subjected to bending moments, and if you imagine a crest like that bending, then the closest analog you have to a test would be a single cantilever bend, because it's fixed at the base of the skull, and then it might bend at the top and so forth, right? So that would perhaps be a good sort of analogy there. But what's important is that if something is bending, you'll have tension on one face, compression on the other face, and in between you will have reduced stresses until you reach a neutral axis. So you don't need as much of the stiff, strong material to basically resist loads in the middle of your bone, of your amphitheater bone. And if you look at the image on the left, you will see very clearly that that is the case. You have thick sections of lamellar bone, which is primary bone. It's a very stiff kind of bone that's on the outermost faces, and on the inside where you have where it's less important that you have stress resistance or resistance to loading, you've got this porous architecture, three-dimensional porous architecture. Anyway, I'm going to go back to the crabs now. That was just a short interlude about composites and biological composites, of which there are many. Anyhow, there was some work I did also on these guys. These are decorated crabs. This one is Tiernia cornigera, and it is a passive decorator. If you see this crab walking around in the sea, it'll usually have lots of macroalgae attached to it. But we saw these crabs and we wondered, how do they passively attach algae to their backs? Because most crabs that you come across have very clean shells, right? So what's going on with these guys? So we basically cleaned it up a bit, removed the macroalgae, and we did some SEM, and we found that there were these like these flowery haystack type structures growing out of the carapace. And if we did some CFD simulations on the surface of those, as you can see on the right-hand side, what we found is that there are these stagnation points that arise on the surface of the carapace. Now, our SEM also showed us that there are diatoms attached to these haystacks, let's call them. So, what we understood then was that these stagnation points are basically holding diatoms and potentially other EPS secreting microorganisms. EPS means extracellular polymeric substances, and they are often in microorganisms very sticky. They are the things that cause biofouling. So yeah, basically, we've got these diatoms that are stuck there, other microorganisms that are stuck there, secreting EPS, secreting this bioglue. And then when you have enough of that, you can probably start attaching macroalgae and that kind of stuff. Now, what is a diatom, you're probably asking? Well, a diatom is an incredible single-celled organism that has a glass skeleton that has a nanoporous architecture, and there is no such thing as a diatom per se, because there are, according to phycologists, about 150 to 200,000 species on Earth, of which about 40,000 have been named, but they come in all sorts of shapes and sizes. You've got cylindrical ones, you've got like long, thin ones, circular ones, you've got weird shaped ones. So diatoms come in all sorts of types of shapes, and the important thing here is that they secrete this sticky glue, and that they form a bioglass skeleton that is one of the strongest natural materials on Earth. It has a strength that was measured at around, I think you find the paper in PNAS, at around 1 gigapascal, which is pretty good for a natural material. Something like bioglass. Anyhow, so when it came to diatoms, I just thought to myself, we need to use these in some way. We need to, we need to gain inspiration from them. We need to use them because there's so many of them and they're all over the place. If I pick up soil from, outside my house, there will probably be some diatoms in that soil. If I go to the sea and I just take a cup of water, there are different diatoms in that water. There are so many of them that, we just thought to ourselves, why don't we go against the grain? Because everybody is anti-biofouling. It's very negative. It's very negative, anti this and anti that. It's like, so I thought to myself, why can't we use biofouling organisms like diatoms positively? Why can't we have pro-biofouling technologies? So what I thought I could do is just start coating natural fibers that could be used in composites with glass using these diatoms. And some of them grew on the surface as well. Some of them didn't. It depends on the type of fiber you're using. It depends on the kind of diatom that you're using. But what's important is that where we did manage to coat the fibers in glass, we improved quite significantly the mechanical properties of the fiber. And normally you cannot coat natural fibers in glass, with glass, because natural fibers have a decomposition temperature of about 300 degrees Celsius, whereas glass will melt at around 1500 or so degrees Celsius. So it's very unlikely. And I think the diatom bioglass is actually melted at higher temperatures still, from what I've understood. So this is a clever way of improving the mechanical properties of the fibers themselves. And we did this also using coralization methods. So we mimicked the, corals. They basically form these hard exoskeletons. So we basically formed using polymer templates and calcium carbonate reactions to create these flowery crystals on natural fiber surfaces. We embedded them into polymers, and we found that we pretty much doubled the strength and stiffness of natural fiber composites by doing this. Now, going back to the diatoms again, because I'm not finished with those guys. The diatoms, I just thought to myself with these guys, if you look at how they are in nature, they tend to cluster within, with their own species. It's like they're very tribal. Yeah, they will just cluster. It's like you got these cylindrical diatoms, they'll all cluster together, and pennate diatoms, they all cluster together. Occasionally you'll see, a bit of intermingling between the species, but it's less common, I would say. I thought to myself, why don't we try to forcefully mix the species? We break this tribal mentality between the diatoms, and we're just like, we make these diatoms attach. So I thought to myself, first things first, let's see if they want to attach by themselves. And we started mixing different species with different shapes, different diatoms with different shapes. And what we found is that they actually tend to attach to each other when they do attach in very specific ways. The pennate diatoms, for example, tend to attach by their tips to the bodies of cylindrical diatoms, whereas they will attach by the tips to each other. So, we're thinking to ourselves, okay, well, this could form the arm and the body of a microbio robot. There are other diatoms that will actually enter into cylindrical diatoms, pennate ones that will enter, and then you'll have a glass-glass interaction, whereas the previous image you saw was glass, biopolymer, glass, biopolymer. So you've got soft, hard like that. Sorry, hard, soft, hard, soft. Whereas in this case, you've got hard, glass against glass. So it's something very rigid, but with a shape that is atypical of ordinary diatoms. And these kinds of things could also potentially be used in microbio robots. This is, again, preliminary work. It is published, but we want to do more now in this area to really, what I would love to be able to do is to create a little man out of diatoms about, yay high, and just have him walking on the underwater or something like that. And walking shouldn't be difficult to manage, because there are diatoms that glide naturally. And that's very interesting stuff. So there's a lot of flexibility when it comes to this, but there's a lot of work to do as well. Another thing that we did with diatoms is I was thinking to myself, well, diatoms, they are nano architected. Yeah, so they have really interesting corrugated surfaces, and cells really love topographically irregular surfaces. If you see cells growing well on a surface, it's often a topographically irregular surface, because the cell can actually cling. So if you have a fibroblast, it will form a star if it's healthy, whereas if it's not healthy, it'll form a ball. And what we found is, first off, we needed to make three-dimensional architectures from these diatoms. And as you can see there on the left, we managed to make these kinds of architectures. You could, of course, subtractively manufacture those as well. But what we did is we accelerated the rate of solid state diffusion between the frustules so that they would actually merge into each other, otherwise known as sintering, so high temperature, high pressure. And then we grew our cells on the surfaces of these three-dimensional architectures. And what we found is that they preferred to grow on our architectures, on these diatom-based architectures, than they did on Sigma-Aldrich nunc plates, which are specifically designed for cell growth. And they grew with perfect morphologies. There was no unhealthiness. If you look at the HEX cells and the pre-osteoblasts and the breast cancer cells in this image, you can see that they have shapes that are typical for those cells. Now, the last thing I want to speak about when it comes to diatoms, another area that I've been working in is in bridge design based on diatoms. This is again, taking inspiration from the beauty of the diatoms themselves in order to make beautiful and aesthetically pleasing bridges. But, I'm not ever really content with just leaving things, like that. I wanted to have bifunctionality, so that even though you have this aesthetically pleasing bridge, that you would also have some mechanical benefits, such as pre-stressing or holding the base in place, using the shell of the diatom and so forth. And these are things that we've been also looking into and playing at. And I remember printing one of these out when I was at MIT a couple of years ago, and it was a lot of fun. We're thinking about trying to have an ant trail go through the bridges and that kind of stuff. But anyhow, geometrical design, as you have noticed, is very important in my life, and it is also important in the lives of bees, as Charles Darwin pointed out very clearly. Bees can make apparently true cylinders and spheres. They never begin one cell at a time, always several. They can judge distance to a certain extent, an absolutely perfect structure, economizing labor and wax. So when you look at these hexagonal structures that bees make, we know that we have already mimicked these. We use these in engineering structures ubiquitously. We've got them in the hulls of ships. You'll see honeycomb structures in plane wings. You'll see honeycomb structures in helicopter rotor blades, and so on and so forth. All sorts of places you'll see these structures. And I'm not saying they're not amazing, but they are absolutely amazing, but they are two-dimensional in essence. If you were to CAD draw that honeycomb structure, what you do is you would basically make one unit cell of a hexagon. Then you would make an array, and then you would extrude this, right? That's the simplest way of, time-saving way of, analogously to the bees in that sense. However, it is, in one sense, a structure that, although it has fantastic lightweighting properties in its out-of-plane axis, and less so in its in-plane axis, it is actually a structure that you can look at and you can kind of predict how it's going to work. Okay, so the question I have is, if you have in nature a three-dimensional architecture. So I am saying that the honeycombs are two-dimensional just because you can essentially extrude them and stuff. But if you look at a lot of other structures in nature, and this is some of the some of the baleen skull work. You look at that image, right? If you were to look at that and try to predict how it would work in the same way that you can with a honeycomb structure, I think you would struggle. And basically, it's these kinds of three-dimensional biological structures that that influenced me and inspired me to start working more with mechanical metamaterials, because I desperately want to be able to predict and understand how those 3D structures work. So if you start looking at, mechanical metamaterials are basically materials that function based on architecture and so forth. So if you make simple, complex mechanical metamaterials, and my PhD student Gabrielis is working on this kind of stuff at the moment. So, I don't know if he's around today, but if he is, you can ask him a lot about this. He can tell you a lot of cool stuff. But basically, you've got simple, complex structures when it comes to engineered or 3D printed mechanical metamaterials. Now, I can understand we were perhaps confined to honeycombs in the past, but we have additive manufacturing technologies, SLS printing, whatever have you, that might allow us to make more complex three-dimensional structures, but we need to understand how they work. So if we go to mechanical metamaterials and we can just basically split the parts apart, we can kind of understand them in parts. However, how they work together is a real challenge. And this is something that Gabrielis is looking into, and it's a lot of fun. It's really, really interesting. And hopefully it will lead us to be able to predict how three-dimensional biological architectures also work. Now, next slide is entitled, and this is, I'm coming on to the last couple of slides now. Thank you for sticking with me. This one's entitled A Simple Life, and that is because, if you look at how biological structures are made, they are typically made through cellular processes, and a cell is a very complex manufacturing machine. Yeah, you've got all sorts of things going on. You've got programmed material in there that's telling this, telling the cell to do this and do that, and to merge these proteins and to design these proteins and to spit them out in the form of silica with a bit of phosphorus in it, and so on and so forth. So these are very complex. These are very complex machines, and we can't easily. I know there are a lot of synthetic biologists who are working out there at the moment and doing a lot of stuff with prokaryotes. But I think when it comes to mechanical engineering and the kinds of manufacturing tools we have, we need to simplify things. And I think a good analogy when it comes to geometrical simplification can be basically taken from the art world. If you were to look at the sky and try to paint the sky, what would you do? First of all, for myself, I would panic because I'd think I can't do this. Yeah. But then if I start thinking, I might think, okay, I'll split it up like an artist. I'll discretize this structure. Yeah. And I'll split it up into lots of pixels. And then if I do that, I can identify specific colors and where those specific colors are located. And in similitude, you could do this with geometrical designs in mechanical or any kind of engineering approaches. You could basically look at your structure and try to ascertain what kind of primitives that are easy to manufacture are the ones that you can use where you haven't lost too much of the functionality that you're trying to achieve. That is one of the big hurdles when it comes to biomimetic design, manufacturing. And on that note, I'm going to leave you with the last slide, which combines both biology and engineering. Thank you very much for listening and for sticking with me for the 50 or so minutes that I've been talking. On that note, I guess I'll pass it back to Kahir.
Well, actually,
Yep, Jonathan is.
Yeah, I'll look after the questions. That was fascinating. Thank you very much. I think we've got a few minutes for questions. If people want to unmute and ask a question or put it in the chat, we could take a few minutes to answer some of those.
Okay, so I got a message from Tom in the chat. Thanks, Parv. Hey, går intressant med slut nu. Okay, so he's one of my colleagues from Finland who basically had to go. That's not a question, unfortunately.
So has anyone got any questions they would like to?
Parvez, it's Crupper here. I have a question. There's one topic that you didn't talk about, which was the sea lice. Can you tell us a little bit about the sea lice and the work that you've been planning to do on sea lice?
So the sea lice, we didn't get that funded, unfortunately, Kuher, but we will keep trying, of course. The sea lice are basically like aliens. If you look at the sea lice and the way they attach to salmon, these creatures are absolutely insane. And I should just quickly point out that the Mahaguru for sea lice is at the University of Stirling. His name is Professor James Braun. So if you really want to know everything about sea lice, please speak to James Braun as soon as you can. But basically, these sea lice, when they start life, they're basically about 0.5 millimeters in length. And, they will detect the wave of a fish, of a salmon, if I have understood. Yeah, the salmon lice specifically. And they will shoot towards the salmon at a speed underwater of 9 centimeters per second, which is ridiculously fast when you consider the size of the sea louse. Now, it will attach initially with an anchor, right? So it'll attach with an anchor like this, and it'll hold on and start, feeding off the salmon. And obviously it's going to grow. As it starts growing, it needs to stabilize itself, because this little anchor is not going to work anymore. So it'll basically have a tube coming out from the front and one from its belly, and it'll have a polymer coming out of this one and a setting resin that comes out of this one. So you've got this 2-part thermoset adhesive that forms, and it creates this thermoset rope that sticks to the surface of the salmon that is so strong that if you pull it off, you'll actually rip off the scale. Anyhow, you got the anchor and the rope there, and that's, then the sea louse keeps eating and it grows a bit more. And then obviously as it's growing more, it needs to stabilize itself again. So this staple gun comes out of its head, and it basically staples its own head into the salmon. And once that's done, obviously it grows to about 2 centimeters by this stage. And then you've got this sucker ring situation. It'll basically cephalothorax, it's called. It'll suction itself onto the surface of the salmon, and it is an alien. I mean, I haven't seen anything like that, but they exist, and they exist on Earth. So
the question from Martin.
One of I have potential there when it comes to developing biomimetic adhesives. A lot of potential there when it comes to developing biomimetic mechanical attachment mechanisms and that kind of stuff. But it's something that we need to study, something we need to understand first. There is a question in the chat now. Let's see, who's this? So this is Martin. Is this
Martin? Yeah.
Hi, Martin. How's it going? So great that you came. Thank you very much. I'm honored to see you here.
Yeah, my question concerns the diatoms, and you've put them onto natural fibers. Now, of course, engineering glass fibers, you've only got to touch them, and perspiration will form micro defects on the surface. So would the diatoms actually heal these cracks in the sense they would bridge them to make them tougher?
Yeah, so actually it's a really good point that you make there, because if you look at engineering glass fibers, their properties are actually lower than they ought to be theoretically, just because of defects. And that is the problem with the engineering glass fibers. However, the diatoms don't seem to have, as far as I know, those kinds of defects. And it's possible that you could actually try to grow diatoms onto glass fibers. However, you would be forming a new composite, because there would be, you'd have the glass diatom, but in between you'd have this sticky EPS glue. So it wouldn't work as a pure glass fiber, but it would be potentially a healing composite. I don't know. I mean, I've never tried it myself, but it is a fascinating idea, and it would be really fun to try something like that.
Just the crack bridging mechanism. If it bonds well enough, and then you can make a super composite with carbon fibers as well, maybe.
Yeah, right, absolutely. Actually, there was a project I had one student working on, master's student in sustainable engineering at Edinburgh 2 years ago, and she was basically adding diatoms to the to FRP matrices to try and toughen up the material, and it had some benefits, but some disadvantages as well. And I think the main difficulty that we had was actually being able to mix the diatoms in homogeneously to the resin. And Cahir will know a lot about this kind of stuff, I think, because he is the composites expert amongst us. But we don't know how we can actually mix these diatoms to resins without without them clumping and that kind of stuff. It's quite difficult.
Maybe one more question before we wind up. I'll ask a question then.
Sure.
It's maybe a bit abstract, but at the moment, the field seems to be full of people identifying incredibly novel mechanisms for achieving different functions, things that no engineer would have imagined. Do you think eventually there'll be a set of principles will emerge, that you, instead of just looking for examples, one will be able to create one's own solutions, one's own materials? Is that far off, or is there consolidation?
Yeah, I mean, basically, I do believe the same. I think, that all of this will probably ultimately lead to a set of, a bunch of different equations that will help us to design better, or to design with a specific purpose that is, bio-inspired. How soon we get there, I don't know, but I guess a lot depends on how much funding you can get in and how many postdocs you can get working on the same topic, right? So, but yeah, I think the potential is definitely there. It needs to be explored. We need to make it happen, because there's so much more I think we can do with engineering if we can, basically get these equations in place and these textbooks written. The way we can just open a textbook and say, aha, that's what I need to do.
Yeah, can I follow up on that slightly?
Absolutely.
Obviously, these structures have evolved over thousands, well, millions of years through evolution. So there's scope here for you to set into progress super evolution, so you can speed up the process, which nature has taken so long to develop. How about that for a challenge?
That is a significant challenge, Martin. I will do my best. That's all I can say. So
well, rather than just studying the mechanisms, to actually go one better, or two better, or three better. Yeah. Yeah,
and that's, I guess, ultimately that's the purpose of mechanical metamaterials, is to develop these design procedures for structures that are based on architecture. But then, take it a bit further. And Gabriel is, like I said, my PhD student. He's doing a lot of that kind of stuff. And there are some, amazing findings that we have made, but I can't mention them here because we're probably going to patent them. But yeah, so in that sense, there is this kind of super evolution that is happening, at least at Edinburgh at the moment.
Okay, I think we've got our value out of our speaker, so I'll say a few words and formally bring the evening to a close. I think when the Institute of Mechanical Engineers was founded, I suspect there wasn't a shadow of doubt in anyone's mind that engineering was a distinctly different topic from, say, chemistry or biology or zoology. And although that's still largely true in industry, I think this evening you've given us a view of a future where all these subjects start to merge into one, where incredible materials appear that push the limits of the capabilities we have today. I think it would have been an interesting talk just to hear about the anecdotes, about the spider farmers and the skipping fish and the climbing lemurs and the snapping shrimps and the diatoms. But what really brought it alive to an engineering audience was the numbers that you were able to produce, the science that came with it. So the 193 megajoules of hardness, or the gigapascal of strength, or in fact, I think this evening we had the highest temperature ever mentioned in an IMechE lecture in Edinburgh, 5,000 Kelvin. I don't think anyone has ever come close to such a large number. So occasionally you hear people expressing the view that we're approaching the limits of what's possible in, say, strength or thermal insulation. But this evening you've shown that we're not even close at any scale, whether it's micro, macro. There's new materials, and with those new materials, you've inspired us about the designs that are possible, the devices and the machines that could create new ways of flying or new forms of armor. I think you started with a quote that said, "Scientists study the world as it is, and engineers create the world that has never been." And you've inspired us all to think about how that world could be. So let's thank our speaker for what was an exceptionally good talk. I don't know whether we want to unmute our microphones or you can use an emoji to react, but let's say thank you very much.
Or a bit of both.
Or both. Thank you very much.
Thanks, everyone, and thanks for the invitation.
Thank you.
So our next speaker will be on the 8th of December, Tuesday the 8th of December, and we're lucky to have Neil Young, who's the Engineering and Technology Director at Babcock Forsyth, who'll be talking about digital shipyards and digital battleships. So the 8th of December, if you look on the My Place, near to, no, sorry, not My Place, near to you. My Place is the VLE, Strathclyde. The Near to You website, you'll get details in a week or so. Okay,
We're open for registration already, even though we don't have the full details.
great. Does the committee want to hang on at the end and we can have a quick catch up just for 5 minutes?
Yeah, no problem. Yeah, that's fine.
Thank you very much, Pravaz. That was excellent.
Thank you.
Thanks, Bavaz.